This is my area of research. If you this is one of the most commonly used biology book called the Cell. And on the cover, you see this image of the cell. And normally one would, if you did not study biology with any lens, with any depth, you would think that membrane is the envelope of the cell. But in fact, if you look at the animal cells, all the structure under microscope is just membrane. The other important component, the DNA and the proteins, are not visible. Well, I'm not trying to say that visible part is more important than invisible part, but I just want to point out that these are the physical system. It's not the single molecular system. It's a collection of molecules into a very, very thin film of about 5 nanometer in thickness, but it can extend to millimeter or centimeter in the other dimensions. So it's a very unique physical system, always in water, make it very, very difficult to study. As a result, that in biology, that even though we know a lot about DNA and proteins, we know very little about the membranes. So that's where I think that physicists can make contribution, because this is such a system that biologists have very little tool to study. So just as the busy slides for the kind of experiment I do in the lab, we use something called micromanipulator system, which can look at how the membrane, which we can produce in the lab, and see how it reacts when the membrane is exposed to proteins. That's how the biological system works. Of course, this is just a model system so that you can simplify to the extent that you can understand what's going on. I mean, usually if you start a cell, something happened, very difficult to say what is responsible for what. But here we can sort of separate the cause and effect, so to deduce the physical mechanism behind the action. And we also, these are the sort of the way to observe the phenomena. We also develop a lot of X-ray techniques to study something going on in such a thin film. I will not go into any details, but we are well known for resolving structure in membrane, which for years and years nobody knows what is the nanometer structure within such a soft, thin film. And we have found the structure of various kind of things going on in the membrane. Lastly, Professor Kiang is interested in using AFM to pull the protein to study the mechanics of single molecule. And she has used something called a very sophisticated thermodynamic analysis to construct the free energy landscape of protein when you pull it to see how it changes then. And another activity of hers is study how the DNA recognize each other. She tethered the DNA onto of chosen sequence to Go particle and study how they interact with each other. From that, she tried to deduce the mechanism of DNA recognition. So altogether, well, our group is very young except myself. I'm the only one who has been here for more than 30 years. I have done biophysics research for 20 years, so I have roughly produced 15 PhD. I just want to give you an idea what is a PhD in biophysics, the result of getting a PhD in biophysics. These are sort of the score so far of my 15 students. The other faculty members have been here only for 5-6 years. They have PhDs, but mostly they just become postdoctors. I have a longer history, so I show you this. And the next speaker will be Dr. Matthew Barrett.
Managing astrophysics.